lamp brush chromosome. This name was given to the chromosomes due to their appearance to the brushes which were used to clean the chimneys of the lamps. Due to their resemblance, they are called as lamp brush chromosomes. These chromosomes are found in the oocytic nuclei of some of the vertebrates like the amphibian, sharks, and reptiles. These type of the chromosomes, they are also found in some of the invertebrates, as well as they are also found in some of the plants. Diagram of a lamp brush chromosome, we can see that there is a central axial region, and within this central axial region on both sides there are the loops. This is a loop which is attached to the central region, and this is the second loop which is also attached to the central axial region. So lamp brush chromosomes, they consist of loops which are attached to the central axial region. Loops of the lamp brush chromosome. One loop consists of one chromatid, or in other words, we can say that each chromatid is in the form of a loop. Size of the loop can vary from 9.5 micron up to 200 micron. Lamp brush chromosomes contains a central axial region. Within these central axial region, loops are formed. Those loops are made up of the chromatids. One loop is on one side of the central axial region, while second loop is on the other side of the central axial region. Here we can see the diagram of a lamp brush chromosome. This is a particular region that has been magnified. We can see that there is a central axis of the chromosome, and on both sides there are loops. This loop is attached to the central axial region, while another loop is attached to the central axial region. This loop has been magnified and has been shown that the loop, when it is attached to the axis of the chromosome where it is highly condensed, at this point the DNA of the chromatid is highly condensed when it is attached to the axis of the lamp brush chromosome. One end, there are two ends of each chromatid or each loop. One end of the loop is thin, while other end of the loop is thick. Here we can see in this diagram that how the loop appears, that one of the loop is thick while the other end of the loop is thin. If we see the loop, this one, we can see that it is thick while on the other end it is thin. So this is thick side of the loop and this is thin side of the loop. Same is with other loops, like here we can see this end is thick while this end is thin. So loops which are attached to the main chromosome axis have two ends. One end is thick while the other end is thin. Conclusion: lamp brush chromosomes are found in the oocytic nuclei of many vertebrates as well as in some of the plants.